Hello students, I just wanted to go over a few of the slides from this week's lecture to make sure that we make a connection between the topics and how they're going to lead to the upcoming topics of cellular respiration and photosynthesis. So for this week we have lecture 9, the working cell. And so we're looking at the cell membrane, energy, and enzymes and how that's going to lead us to metabolic processes such as cellular respiration and photosynthesis. So we have the fluid mosaic model of the cell membrane. We went over the cell membrane earlier in the semester with last unit's material. We didn't really cover the function of the cell membrane. We just quickly discussed its overall function of being a barrier between what's outside the cell, that's called the extracellular matrix, and what's inside the cell, that's the intracellular matrix, intracellular environment, I should say. But we didn't discuss the details of its structure, and one of the themes in biology is that structure lends to function. As we see here with the image of the cell membrane, its function is very wide, it's varied. So it has several functions besides just being a physical separation, which is a very important function in itself, but there are other functions to the cell membrane. So starting with the structure of the cell membrane, it's made up of phospholipid bilayer. Bi means two, so two layers of these phospholipids. And we looked at phospholipids when we were talking about macromolecules. They are under the category of lipids, which is one of the four macromolecules of life. So we have lipids, very important aspect of lipids in this example, because it makes up the border of every cell in your body. So we need these phospholipids. We have the phosphate heads, which are hydrophilic. They like water, they interact well with water. And then we have these lipid tails, two lipid tails that are hydrophobic, that repel water. They don't interact well with water. Again, these are carbon chains of carbon and hydrogen, carbon and hydrogen. So we have the phospholipid bilayer, which is the main component of the cell membrane. Embedded in the cell membrane, we have these proteins. Some of these proteins are integral or transmembrane, they cross the whole membrane, and some of them are peripheral, so they're just attached to one end of the membrane, whether it's facing intracellularly or extracellular, they're peripheral, they don't cross the whole membrane. So considering these proteins are either crossing or not, we could see that they themselves will have a different function as well, which we'll see later. Located on the outside of the membrane, facing the extracellular environment, we have carbohydrates. So carbohydrates are either attached to the lipids or they're attached to the proteins. And they also serve a purpose. Usually this purpose is for cell recognition or communication. The proteins embedded are going to be utilized for transport, perhaps, or communication. And then so we have the phospholipid bilayer, the integrated proteins, and the carbohydrates only located externally. Lastly, embedded within the phospholipid membrane, we have cholesterol molecules. So these cholesterol molecules are going to help with that fluidity of the cell membrane. The plasma membrane has these proteins with different functions. Remember that they are either peripheral or integral. And so some of these functions include structural support. So that if they're peripheral, they may have anchored cytoskeletal proteins that are going to help with maintaining that shape of the cell. We have recognition proteins. So all cells have a way to identify themselves to other cells. So we have our immune system, for example, surveys all our cells and is able to recognize which cells belong to self, the same organism, and which cells are foreign. And so this is possible because cells have these proteins that act as identification markers. Then we have some proteins that act as communication structures. So here we may have a hormone, which is a chemical messenger, has a receptor on a protein embedded in a cell membrane. And so that hormone chemical messenger attaches to the receptor, that protein on a cell membrane, and that integral protein transmits the message to inside the cell. So this is one mode of communication between cells and to our cells between our tissues to other tissues. And then transport, proteins are very essential in transport. The cell membrane is a physical barrier, meaning that it's selectively permeable. It allows certain molecules to go across the cell membrane, and it prevents certain molecules from going across the cell membrane. And so certain molecules need to go inside the cell membranes. For example, glucose, the glucose that you ingest. We went over the digestive system chapter in nutrition. You eat, the molecules are broken down into the basic components. So carbohydrates are broken down to glucose. That glucose is needed inside the cells because that glucose is a chemical, a molecule that has chemical energy. And that chemical energy is needed for functioning of the cell, which is the topic of next week's topic, which is cellular respiration. So that glucose needs to be transported from outside the cell to inside the cell. So a lot of proteins are going to act as facilitators, carriers, to facilitate that transport. Carrier proteins are doing that carrying of the molecules across the cell membrane. So all carrier proteins are specific to a single substance. So for example, glucose being transported across the cell membrane are using very specific transport proteins, and they're called GLUT transporters for short, glucose transporter proteins, or GLUTs for short. So this movement of proteins or molecules, I should say molecules, because in the case of glucose, it's a carbohydrate being moved across the cell membrane. That's driven by diffusion. The molecules are going from an area of higher concentration extracellular fluid to an area of lower concentration. It is a passive movement because it does not require energy. The energy is in the location of the molecule and the concentration of the molecule. So that high concentration of glucose in the extracellular environment will drive the movement of glucose down its concentration gradient to inside the cell. And again, this is facilitated by a specific carrier protein. However, some molecules need to be moved across the concentration gradient. And if that's the case, moving molecules across a concentration energy will require energy. And that energy comes in the form of adenosine triphosphate. Taking a closer look at the cell, we have in blue on the left the extracellular fluid or matrix, so that's outside the cell. We have the cell membrane creating that barrier between what's outside the cell to what's inside the cell. So on the right-hand side, we have the intracellular environment inside the cell, and we could see that there are a number of molecules inside and outside the cell. And from this image, we could see that the A are, they could be amino acids or proteins. They're negatively charged. And then we have potassium, positively charged. We have molecules of sodium chloride, salt, that are bound as an ionic compound. And then we have some free-floating chlorine atoms. So overall, the net charge of inside the cell is negative. If we look at the extracellular environment, the net charge is positive. So we have salt molecules, sodium chloride, and a lot of sodium chloride on its own that are positively charged, and some potassium, etc. These are just an example of some of the molecules you find inside the cell and outside the cell. So generally speaking, inside the cell is negative, negatively charged proteins and other molecules, and outside the cell is positive, and that positive is really driven by that positively charged sodium cation, to use the terminology we learned in chapter 2, chemistry chapter. Now, this arrangement, net charge positive outside, net charge negative inside, is not at equilibrium. And it is maintained at this steady state of disequilibrium. And it's necessary for this steady state of disequilibrium to be maintained. It's one of the variables that needs to be maintained. So part of homeostasis is a maintenance of this disequilibrium of charges inside the cell and outside the cell. The steady state of disequilibrium negative in the inside the cell, positive at the outside the cell, is accomplished by a very special integral protein called the sodium-potassium pump. So the sodium-potassium pump shows you an example of active transport. Active transport, again, is when transport needs to occur against the concentration gradient of molecules. And active transport denotes that you need energy, an input of energy. That input of energy comes by the means of adenosine triphosphate, or abbreviated ATP. So we have ATP here shown as the pentagon attached with three little balls are called phosphate groups, three phosphate groups. And so we have that molecule of ATP providing that energy to move molecules across a concentration gradient. In this case, for the sodium-potassium pump, we have three molecules of sodium are going to be moved from inside the membrane to outside the membrane. And two molecules of potassium are going to be moved from outside the cell membrane, extracellular environment, to inside the cell, to that intracellular fluid. So here we have the cytoplasm. And this potassium pump is continuously working to maintain a constant disequilibrium of charges. And we have more sodium outside the cell, more potassium inside the cell, and an overall negative charge inside the cell, and an overall positive charge outside the cell. This is very important in maintaining that homeostasis, that is disequilibrium. It'll lead for the ability of cells to generate electrical signals for communication. It's possible for other molecules to move across the cell membrane. And so one example of how this is facilitated is if we look at secondary active transport. So you can look at this animation for active transport. It shows you exactly how the sodium-potassium pump functions and how ATP plays a role in that function. So primary active transport drives secondary active transport. The sodium-potassium pump is actively moving sodium outside, so it creates this concentration gradient of sodium outside the cell higher than inside the cell, seen by this arrow. And so since there's a higher concentration of sodium ions, cations outside the cell than inside the cell, that's potential energy. So it's potential energy in its location and its concentration. This potential energy could drive the movement of other molecules against the concentration gradient. So for glucose, we have low concentration outside the cell, extracellular environment, and higher concentration inside the cell. So in order to move glucose inside the cell, energy is required. Now this energy doesn't come directly from ATP. It comes indirectly from ATP by the location of the sodium ions, the higher concentration sodium ions. So the sodium ions want to move down the concentration gradient. We have these transporters called symporters. They're going to transport two different molecules same direction. So here we have sodium is going to be transported alongside glucose into the cell. And the energy for this transport comes from potential energy of the sodium concentration higher outside the cell than inside the cell. So that's an example of secondary active transport. There are other transporters that use primary active transport to transport molecules besides sodium across the cell membrane. So we have one example here, the proton pump. It's called a proton pump because it's transporting hydrogen ions. And if you recall, hydrogen ion, it has one electron outside its orbital shell. And so the first orbital shell either needs to have two electrons to satisfy the octet rule, although since it only has one electron, it's only meeting that one in the first orbital shell that can occupy two electrons. For hydrogen, it's just as easy to gain an electron to lose an electron. So since it's lost an electron, these are known as hydrogen ions, cations. And really, without an electron, it's just a proton, because the number of electrons always match the number of protons. So it's just a proton. So these are called protons as well. So we have a proton pump. Similar to the sodium-potassium pump, it uses energy in the form of ATP to pump hydrogen ions across the cell membrane and creates a concentration gradient of hydrogen ions on one side of the membrane, higher in one side than the other side of the membrane. So again, that energy comes from ATP. ATP has 3 phosphate molecules. When one phosphate molecule breaks off, that releases energy, and so it becomes ADP, adenosine diphosphate, plus its inorganic phosphate, and that action drove the movement of hydrogen ion or protons across the cell membrane. So now there's potential energy in the concentration and location of the hydrogen ions, and this concentration, this potential energy, is going to drive the movement of, in this case, another type of sugar, sucrose. So here we have sucrose and hydrogen ions are going down the concentration gradient. Sucrose is being transported across the membrane. This again is a symport, and another example of active transport and secondary active transport. Now, these are important concepts to understand because this shows you how cellular respiration will function later on, one of the steps of cellular respiration. And so it's important to understand the movement of these molecules, the role of ATP, when we talk about a very important metabolic process that occurs in our cells, and it's essential because without it, we will die. And so make sure that you understand this movement of molecules. If you have any questions, of course, email or ask in the class discussion. Energy, ATP, essential to life. We looked at this very first lecture, right? Essentially, energy is derived from the sun. All living things require energy. Our organisms, right? Living organisms, about the characteristics of living organisms, one of them is that they're highly ordered. They're complex, very organized. And this organization, this complexity requires a lot of input of energy. And so we have solar energy, we have heat and solar energy. Producers, photosynthetic organisms, are able to capture that solar energy and convert it to chemical energy. That's in the process called photosynthesis. Now, consumers, heterotrophs, are not able to produce their own chemical energy in the form of glucose, so they need to consume that energy, so to speak, that matter that contains that energy. And so animals, consumers, ingest matter and so are able to transfer the energy, the chemical energy in the glucose, to the ATP molecule. And then the ATP molecule goes and powers other reactions as we have previously seen in the previous slides. Okay, so bioenergetics is the study of energy flow through a living system. Types of energy, we just saw examples of potential and kinetic energy. The potential energy was in the location and concentration gradient of the sodium in one example, the hydrogen ions or the protons in another example. That's potential energy. But when they're moving down their concentration gradient and sucrose is being moved into the cell and glucose is being moved into the cell, that's movement, so that's kinetic energy. Energy of chemical electrical gradients across the plasma membrane. And so chemical energy, energy stored in chemical bonds, that's potential. So when we ingest that food and it's being broken down, that's going to be an example of the release of energy, especially when glucose is broken down further from glucose, hydrogen, and oxygen, right? Glucose is C6H12O6, and that there's energy in the bonds of the carbon and the hydrogen. That's potential energy. So when we eat, we have that chemical energy, potential energy, and we utilize that potential energy, convert it into kinetic energy that gives us that boost. And so we have kinetic energy. Likewise, with law of thermodynamics, we have solar energy being light energy being converted to chemical energy through the process of photosynthesis. In these processes, metabolic processes, energy will be lost in the form of heat. And so heat is its most disordered form of energy, so it increases entropy, disorderedness. All right, so looking at an example of energy and its conversion, we see here uh, octane from gasoline and oxygen. Again, we put gas in the car because that's its fuel. That fuel, through combustion, changes that chemical energy to form a kinetic energy and causes the car to move. Okay, so it's energy conversion in a car. The waste products of this combustion of gas is carbon dioxide and water. So then we have CO2 being emitted into the atmosphere. But if we look at what's happening in our cells that have mitochondria and are able to do aerobic cellular respiration, we have glucose. We ingested the molecule, it was broken down by our digestive system, it was transported across our GI tract, the intestines, made into our bloodstream. It's being transported in our bloodstream to be delivered to our tissues and our cells. We have that glucose and oxygen that we inhale. So the glucose and oxygen makes it into the cell. So this is one cell. And through the metabolic process of cellular respiration, ATP is generated. So that energy from glucose is being transferred to the ATP molecule. And we have ATP that could go on and provide the energy for cellular work, such as that transporting of molecules, right? That sodium potassium pump needs a lot of energy. You stop that pump, you stop life. And so the waste products of cellular respiration is carbon dioxide, and you can't see here, but it's water, carbon dioxide and water. So really, the gasoline burning is very similar to cellular respiration, what's happening inside our cells. Now, how is it that we don't have this fire inside of us, so to speak? Okay, well, cellular respiration is a very, very controlled metabolic process. Okay, it's very controlled, but it is a combustion. It's a chemical combustion process. And cellular respiration is a 19 enzymatically catalyzed reaction. So it is a very, very controlled reaction. It's not only controlled by enzymes, and we'll talk a little bit more about enzymes shortly, but it's also controlled by the way the eukaryotic cell is arranged. So the eukaryotic cell, again, we have compartments. You have organelles, right? The mitochondria is a double membrane organelle, and the processes that occur outside the mitochondria in the cytoplasm are physically separated from the processes that occur inside the cell, inside the mitochondria. And so for cellular respiration, we see it as a three-step process, but really those three steps, each of those steps are several steps that are separated physically, but that are also controlled enzymatically. So a metabolic process pathway is a series of reactions catalyzed by multiple enzymes. So cellular respiration, next week's topic, is a metabolic process catalyzed by multiple enzymes. So a brief overview of a metabolic process. We have the substrate in green. We have the enzyme as the little Pac-Man figure. We have the enzyme 1 binding with the substrate. We have an intermediate substrate. Then we have enzyme 2 acting on the intermediate substrate. We get a second intermediate substrate. Enzyme 3 acting on the second intermediate substrate. And then we get an end product. So just a quick overview of how we're going to be having a substrate. And our initial substrate for cellular respiration is glucose, C6H12O6. Let's write that down. C6H12O6, glucose. And then that's going to be transformed into or changed into different substrates, but being acted on enzymes, where we're going to get the end product. And that end product for cellular respiration, I'll draw a quick arrow here. It's not going to be a balanced equation, but we're going to get our metabolic product, carbon dioxide, water, and what we're mostly interested in, ATP, and heat. We'll always get heat as a last form of product, although really I shouldn't say that, because for our bodies, that heat is what's maintaining our body temperature, that heat byproduct of cellular respiration. But here's the ATP that cellular energy that we need for all other reactions. And so that end product, ATP, in this for example, the end product inhibits the whole process. So we call that end inhibition, end product inhibition. Just a quick overview. We won't go into details of metabolic processes. The cellular respiration, we're going to go over main steps. We won't, your biology for non-majors, so you don't have to memorize the different enzymes or the different products from each of the steps. But we do need to understand the overall process of cellular respiration, what the main three steps: glycolysis, the citric acid cycle, and electron transport chain. What we start with, what we end up with. And again, we're starting with glucose. My circle was a little off there, and we're ending with ATP. So enzymes are very, very important. If you recall, when we talked about macromolecules, we have 4 biological molecules of life. We just discussed lipids earlier. We have proteins, and proteins are essential. They have a wide variety of functions. We just saw a few of the functions in this lecture. We have them acting as receptors for cell recognition. We have them acting as transporters, carrier proteins. We have them acting as enzymes. So what are enzymes? Enzymes are proteins, and they're made up of amino acids with a specific shape, right? The chain of amino acids, it's a primary structure of proteins that folds up into beta sheets or alpha helixes. That's secondary structure of proteins. And that changes into a 3-dimensional globular shaped protein. That's its tertiary shape, and it can move on to a quaternary shape. So the final shape of the protein determines its function. And so we have enzymes being a special type of protein that accelerate chemical reactions. What that means is that they make chemical reactions go faster. They're biological catalysts. Without enzymes, reactions would take place, but too slow to be compatible with life. So we have enzyme, we have lactose, for example. Lactose is glucose and galactose. It needs to be broken down. That's a chemical reaction. We looked at this earlier in the semester. And so there's energy input. That's called the activation energy without the enzyme. If you have the enzyme, lactase, so we have lactase in green come in, it lowers that activation energy. And by lowering this activation energy, it's going to be a biological catalyst and make the reaction go faster. So we get the end product, glucose and galactose. And substrate specificity, enzymes are specific for specific substrates. Okay, so the 3D shape of an enzyme and reactants or substrates determines its specificity. The enzyme has a particular area where it reacts with the substrate called the active site. And the way that they react is called the induced fit model. So it's a lock and key model modified. The enzyme substrate fit together like a lock and key. However, it's modified, it's induced fit because as the key is coming in, the lock is changing and modifying itself slightly to make sure that the key fits. That's what it's induced fit model. Enzymes can catalyze a variety of reactions, but the enzymes are very specific to their substrate. Enzymes, once they do their job, they're unchanged at the end of the reaction and could go and carry on that same reaction over and over and over. Enzymes are important. They play different roles. So some examples of enzymes include catalase, hydrogen peroxide broken down to water and oxygen. When you pour hydrogen peroxide on a cut, for example, to disinfect it, all the bubbling is the oxygen that's produced with the breakdown of the hydrogen peroxide. Starch breaks down to maltose, and the activity of amylase. We might have discussed this with the nutrition chapter where your saliva contains amylase. That's the enzyme that helps break down the starch, and the starch, another name for starch, is amylose. Lipase breaks down fats, and then pepsin, it's an enzyme we did mention, it's in the stomach, works best at low pH, breaks down proteins. Enzyme regulation, so there are environmental factors that will regulate the enzyme, and there are chemical factors that regulate the enzyme. So enzymes can be regulated by modifications to temperature and or pH. Recall that we looked over digestive system chapter. Our saliva, the environment in our mouth, is near neutral. So amylase works best in near neutral pH, as opposed to pepsin. Pepsin's in the stomach. The stomach has a low pH at the very distal end of the stomach. In that low pH of 1 or 2, will destroy most proteins and enzymes, right? But pepsin works best at low pH. Amylase would not work in the stomach, but pepsin does. Pepsin will not work in the mouth, etc. So modifications, temperature, or pH. Products of molecules. Well, if we're talking about our enzymes inside our body temperature is 37 degrees Celsius, 98.6 Fahrenheit. So we know that that's the optimal temperature for our enzymes to function. Production of molecules that inhibit or promote enzyme function. Inhibitors, they compete for that active site, etc. Availability of coenzymes or cofactors. So looking at some of the regulations of enzymes, we have temperature. So I encourage you, you have the slide answers, but I encourage you just to think about this and try it without looking. You have the temperature, so you could go from 0 to 100 Celsius. 100 is the boiling point of water, and 0 is the freezing point of water. And you have a range in between, right? We have 50, and I'm going to put about 37 right here. So that's our body temperature. So if we are talking about amylase, we know that's going to work best. So the product formation per unit minute reaction rate. So how much product we get? We're going to get a lot of product if we're talking about amylase. However, if we put the same amylase to work on amylose, on starch, and break it down at 100 degrees, well, that's the boiling point of water. That's going to denature enzymes. So that means that we're going to have very little product because the enzyme's denatured. It's destroyed. You change the shape of the protein, you end its function. You destroyed its structure, you end its function. You destroy its function. 0, you basically stop molecules from moving. You're reducing that kinetic energy. So really, you have stopped the reaction. So we're going to have very little product. So then your graph, you have a point where there's a lot of product and a point that the product formation or product declines to a point that there is no product being made because the enzyme's not present. So I encourage you to go through these and see how each of these factors affect the enzyme substrate reaction. We have pH. Again, you could look at an example, or you could, estimate which pH would be optimal for an enzyme. Don't forget that the pH scale goes from 0 to 14. So you do have a range. Substrate quantity, that means you add more substrate, but you keep the enzyme concentration constant. How does that affect the formation of product? Then and we have what I previously mentioned, just that according to the different parts of our body, we have a different pH, and that means we're going to have different enzymes that have different optimal pHs. Enzymes are also important commercially. They are used in laundry products, so they help break down proteins. So you get a stain on your shirt, that stain is a protein. So we could use enzymes to break down the protein and clean your clothes. Pectinase breaks down the substrates in apple cell walls, enables greater juice extraction to make apple juice. Lactase, if you're lactose intolerant, then you might be buying lactose-free milk. Well, that means that they added lactase, the enzyme, break down the lactose, and therefore you have lactose-free milk. So enzymes very important. They're involved with our next topic, cellular respiration. But again, the topics we've discussed for this lecture, the cell membrane, products that or molecules that go across the cell membrane, the structure of the cell membrane, the cell and cell structures are all information that you needed to understand metabolic processes. And so next topic, cellular respiration, photosynthesis. Very important that you understand this topic and that you keep in mind previous lectures and information that you've learned so you could better understand the importance of cellular respiration and photosynthesis, our next upcoming topics. So, if you have any questions, email me, or you could write down your questions in our Q&A discussion forum. Thank you. Have a good week.